Good evening. Welcome to the public lecture organized by the Institute for Mathematical Sciences. This evening, my name is Chang Chitat. I am the director of the Institute. This lecture series is sponsored by the Ng Kong Beng Memorial Fund and the IMS, Institute for Mathematical Sciences, as we call it, holds several public lectures each year. And we are very pleased that this evening I got Professor Benoit Perthame from Sorbonne-Université to give the public lecture. Professor Perthame graduated from the École Supérieure in Paris and received his habilitation under the supervision of Pierre-Louis Lyons. 1987 or 88, around that time.
87.
Right. Yeah. And his research interest is in the applications of nonlinear partial differential equations to mathematical biology in relation to multi-scale aspects of chemotaxis and self-organization and movements of cells and bacteria. He's also interested in neural computing, tumor growth, as well as evolution. So all the applications of mathematics to these areas. Professor Perthame has got a very distinguished career. He was an invited speaker of the International Congress of Mathematicians in 1994 at Zurich, and then again in 2014 as a plenary lecturer at the International Congress that was held in Seoul, Korea. And he has received many, many awards. I just name a few. He received the Blaise Pascal Medal in 1992, the INRIA Prize from the Academy of Sciences in 2013, I believe. Or was it 15? And in 2017, he was elected to the French Academy. That's, I think, among the most prestigious, distinguished recognition of the achievements in France. And he is here in Singapore as a distinguished visitor to the IMS program called Quantum and Kinetic Problems on modeling, analysis, numerics, and applications. And he is, I should also say, on our scientific advisory board. And we're very happy to have him as a board member. So this evening, he'll give a public lecture with the title called Some Equations from Biology. So please welcome Professor Perthame.
So thank you very much for this introduction and for the opportunity to visit this marvelous university for several weeks. So as you mentioned, my recent interest has been recently to understand how mathematics occur in biology. And of course, one of the places mathematics is the most importance in biology is signal analysis, data analysis, and because biology now is able to record huge amount of data, which has been the major change in biology in the last 40 years. The access to data before it was extremely complicated. To have movies, they have images at a different scale, especially at the molecular scale. And partial differential equations have nothing to do with signal analysis. The idea of partial differential is to give a meaning to the signal you receive, to explain why you observe what you observed after you have been doing the analysis of the data. And I would like to illustrate that on a few examples coming from biology. But before that, maybe I would like to recall how important partial differential equations have been in physics. and more or less the status of physics. All the area of physics now are recognized by equations, which bear the names of the people who invented them and who showed how they are important. So one of the questions many people ask me, are there equations? Are there fundamental principles in biology? Some people say, no, mathematics is good for physics, but biology is different. There are no, it's not a good language to describe biology, which is too complex. Alan Turing was one of the most, what the most important in modeling in biology, was saying science is a differential equation. So let's take the simplest equation. I think everybody who has done science in his studies have seen at some point the Newton's equation, what we call the principle, the fundamental principle of dynamics. These equations describe the dynamics of particles. You throw particles, particle in a force field. This is what will describe the trajectory, so position and velocity. So everybody, I think, have seen that kind of equation. Who have never seen that kind of equations? So that's fine, so far. So now if you take many, many particles, you arrive to fluids. So another area which has been written in terms of equations which bear the names are the Navier-Stokes equations. So this is Euler, this is Navier, this is Stokes. So these people, these three persons, wrote what are called the Navier-Stokes equations, which are used to compute the motion of a fluid, so which you describe by the velocity of the fluid, an incompressible fluid, so by its velocity and pressure. Yeah. This is that is the most example of part of physics where we know all the equations, their validity, and which are used every day in our life, even if we don't know it. They are used to predict the weather, for example. Ludwig Boltzmann is known to explain that finally there is a connection between Newton and Navier-Stokes because what we think of the fluid is in fact the collection of molecules colliding together. And if we were able to see the atmosphere around us, it's made of molecules. And of course, Boltzmann, 1872, had the difficulties for his colleagues to admit that this is the point of view that you should use. And if you do that, not only you create a new area of physics, which is the physics of dilute gases, those you find in the atmosphere when you compute the trajectory of a satellite in the upper atmosphere, or when you send a satellite in the upper atmosphere. But he also created a new kind of equations where, by opposition to Navier-Stokes, where the velocity only depends on space, now the distribution of molecules is referred as space and their velocity. So now you describe the dynamics in dimension 6, 3 for space, 3 for velocity, plus 1 for time. And so conceptually, you arrive to a new kind of equations, which are called the Boltzmann equations, the kinetic equations, which describe simply how particles are moving and how particles are colliding. And this is what we call now the phase space. Of course, physics is used to use phase space in the phase space in many areas. What mathematicians can do is to prove that particle systems, if the number of systems is big enough, will converge to the Boltzmann equation. So they collide, and then you arrive to a density of particles. This is discrete. You have a number, capital N, of particles. You go to a discrete level, to continuous level in the phase space, and this is when N goes to infinity. And here you have a number, which is a ratio between collision and transport. And if this ratio is very small, I mean, it's very big, this kappa converges to 0, we know that the Navier-Stokes equations can be derived from the Boltzmann equation. So this is what we call in mathematics a multiscale analysis. From a scale which is the size of the molecule, you go to the scale here of trajectories between molecules, and then you go to the scale of the macroscopic scale that which is our scale in this room. There are many more equations. So the most famous is called the Laplace equation or or Poisson equation. Just describe the potential if you have a density of charge F. The oldest equation, the oldest PDE ever written by human beings is the D'Alembert equation. It was written by D'Alembert. It is a wave equation. It was used to describe the motion of a slope when you move it. The Fourier equation is the beginning of the 20th century, describes the diffusion of heat in a media. So it is used in engineering very commonly now, but all of them, but they are used commonly. Maxwell is famous for the Maxwell equation. Electromagnetism is described by Maxwell equations. And if you think of quantum mechanics, now we are at the beginning of the 20th century, the quantum mechanics is described by the Schrödinger equation, which is close to the Fourier, but there is an I. We are in the complex field now. So you see electrostatics, wave propagation, heat propagation, electromagnetism, quantum mechanics, all of them have their equations, they have their names. I have tried to gather the names of the equations I know. And of course, I got this big file. I'm certain that I'm forgetting some of them. So if you see an important equation which is missing, just tell me at the end of the talk so that I can include it for next time. So the question is, and in biology. So when thinking of this, the first name which came to me is the name of Lotka-Volterra. I think Lotka-Volterra are the most famous equations. You find them very early in calculus for when you want to find examples. They describe the dynamic of a small population of small fish, S of t, and of predators, so big fish, which eat the small fish. So this is a term which explains that the number of small fish will decrease by predation by the predators. And these predators create a biomass of predators, which is here. The small fish reproduce because they eat smaller fish again, or phytoplankton or plankton, and the predators will die if they have no small fish to eat. So Lotka-Volterra, these equations were written in the 1920s. And by the way, Volterra used that to explain the proportion of small fish compared to big fish in the Adriatic Sea. He was a professor in Trieste, in Italy. But this name, Lotka-Volterra, now has become a very generic name for a large class of equations that we are using. So in the modern language, you don't want only to speak about two kind of species. I mean, these small fish, certainly they have many different small fishes with different sizes. And these prey, there are certainly predators. There are many predators with different sizes. So you would like to have what you call a phenotypical trait, X, which represents the size of the fish. So the different species of fish in the Adriatic Sea. But this could be very different. The phenotypical trait, it's amazing how biologists have imagination to understand how populations interact with their environment. It could be the color of the animal. If you have different colors, it can be predators can be more efficient on you, or you can have it might be easier to find to mate. So color could be them. There are many examples. Cannibalism is a parameter. The level of cannibalism in many species can be one of the parameters you use. And then your density of individuals, the number of individuals now is not a discrete thing between two types of species. It becomes a continuous object depending on this phenotypical trait of this size, for example. And then you have environment that would be described like that. And what we write is simply that the change in number of individuals is proportional to the population with a growth rate which depends on the environment, of course. There are many, if the food is good, the population will grow. And we know since Darwin that there are mutations, not there are variations in individuals. The mother of type Y can give birth to fish of type X. And so you describe it by this kind of mutations. This is the modern version of Lotka-Volterra equation, since if you see that from the point of view of Darwin selection, because you input mutations and you input here the mathematics of selection, because you select those who will grow faster. What is surprising is that if you put these equations in a computer and compute the solutions, what you find is this kind of distribution of the trait. So here you have the trait, here you have time. Initially you put a certain type of individuals, and what you observe is that the trait will evolve. Maybe there will be a branching, and two types of population will coexist. And maybe further, you get another branching. Now you have 4 types of individuals which can survive together, but one of them will disappear by the pressure, by the selection pressure of the others, and finally you get only 3. This is exactly what Darwin, this drawing is taken from the book of Darwin, The Origin of Species in 1859. So you see that he already had this idea that you have this kind of branching, which was written. He's the first one to write this, and you find now that this is what a mathematical model will give you. What is represented in this picture is the same thing, but when you write a PD, an equation like the one we have seen before, there is always stochastic, a probabilistic interpretation, where now you think of individuals, a finite number of individuals, which will reproduce with a random rate, which will die with a certain random rate, which will have mutations with a certain probability. And all this make that behind these equations, you have always a probabilistic interpretation. One of the rules of mathematics is to go from this to this and explain the connection. And so what you see here is what we call a Monte Carlo simulation for the same process. So I think this is one of the most famous equation. It is not the oldest equation in mathematics. If you want to find the oldest equation, partial differential equation written for biology, you have to go to epidemics, the spread of epidemics. And again, we have very many models for the spread of epidemics to explain how, in infected people will encounter susceptible people to create new infected people. This is something which has been known for a long time. In France, there is a paper by Daniel Bernoulli, one of the brothers Bernoulli, who built more or less modern analysis in Switzerland, and they published this Mémoire de l'Académie Royale des Sciences. So when you mentioned that I've been elected at the Academy of Science, in that time it was called the Académie Royale of Science, but of course we changed name. Not only we cut the head of the king, but we also changed the name of the academy. So this is before. So for those who know how to compute, to read this number, this is before the French Revolution. So this equation is known is very easy. It's an equation that also you find in calculus. What is not known is that if you input periodic forcing because of seasons, which occurs in Europe more than in Singapore, then the solutions become chaotic. So it's extremely difficult to predict the flu in Europe just because of this chaotic effect by the dynamics of this system, which synchronize or not doesn't synchronize with the periodic seasons. So to improve the model, Kermack and McHenrick proposed to be more accurate and wrote what I would call the first equation of mathematical biology. So what they wrote is that finally, it's more or less, if you see, it's in the spirit of Boltzmann. So now they say the number of infected people should not be labeled only by the time, but also by age in the infection. Because depending on your age in the infection, you will have not the same probability to propagate the infection. Some disease, at the beginning, you are not infective at all. Some of them, you are infected at the beginning, not when you have the symptoms at the end. So he says you need to recover the age of the infected people. And to do that, you have to write that the age after infection progresses like the time. And so he writes, like Boltzmann, an equation saying that your age is increasing with time. And of course, then you have all the other terms explaining how a susceptible will interact with an infected with a rate which depends on the infection, the time, and of course, you need these equations come with boundary conditions. You need something to say that you are infected when you have met susceptible. Susceptible have met an infected. So this is certainly the first equation which was written specifically for biology, have no connections with physics. So nowadays we have many tools to handle these equations. I mentioned this here in labs which are working on epidemic propagation. They can make global computations on Earth, on the main city on Earth to see how the disease will propagate. So this is the second example. Epidemic propagation is one of the classical examples where equations arise in biology, and they have a name. The third example I wanted to show is neuroscience. So neuroscience is also a place where mathematics has played a role, for the reason that basically neurons are electrically active cells. And so when you want to describe the dynamics of neurons in terms of their potential, you are back to electricity, so to things that Maxwell settled in the 19th century. And here, I think the name which is the most famous is the name of Huxley. They won a Nobel Prize for writing the equations which bear their name. Maurice Lécart is quite famous also because they are also, as Huxley, they were able to fit experiments on their model and give a complete description. Here it's for the muscle, here it's for the axon of the nerve. The equations that they wrote are these equations. So you have a potential, which is a membrane potential, and you have the gating variable, which are the opening of ionic channels, for example, of synaptic connections in networks. And these are ODEs, which are quite complicated because they describe the dynamics of the potential of neurons. And if you have learned that, you know that neurons are characterized by the fact that they are able to have spikes, which means that their potential, if you are at rest, at some point can grow very quickly, reach a high level, 70 millivolts, and go back to rest. And if you have excitation, if you put a current, which of course you can do in a laboratory, then these spikes come again. And so you have this periodic activity of this neuron in that case, which you see here. So this level where it's below is called hyperpolarization. So more or less all the equations I've shown before, they are adapted to, they exhibit this kind of dynamics, so that's the reason why they have been considered as good models. But not only that, they also give the correct scales and the correct shape of the spikes. Very quickly, a physicist, so I said that probability is very important. I would like also to insist that physics is very important because behind electricity, for example, all physics is there. So physicists decided that it's not enough to describe a single neuron. What you want to describe are networks of neurons. And if you want to describe a network of neurons by this kind of equations, then you will face very complicated problems because we'll go very quickly in very high dimensions. You want to connect millions of neurons together. This is a very small network, will be millions of neurons. So to connect all of them and see their dynamics is quite challenging. So they said, okay, it's better to simplify the model, and they invented what is called the integrated fire model. In fact, it was not invented by a recent physicist. It goes back to the early 19th century also. Louis Lappic was the first one to propose that to simplify the dynamics by simply ignoring the variable, the ionic channels, and creating a much simple equation where you see again a Brownian motion coming in, so randomness, where he said when the potential arises, a certain firing potential, I will go back to reset. I will not try to describe all these dynamics, which is complicated. When I leave the rest, the equilibrium, then suddenly I make a reset. So I consider that this spike doesn't exist, and I go directly from there to there. That's what Lappic was proposing. Louis Lappic is not very well known. I don't know if you heard about him. His brother is very well known. Charles Lappic was a very famous painter who invented more or less the use of colors in that time of the history of painting. So if you simulate that, with a very simple equation, what you see is that your neuron has a dynamics because you have randomness here, you have noise, you have some randomness, but the dynamics is given by these spikes. So these spikes, you don't describe it. You just go directly from the firing potential to the reset potential, and you continue. And so the physicists, and some names are here, consider this is much simpler to write networks like that because the equations are very simple. It's a single equation, and they know how you should find the parameters on the equation, because they knew that if you have an experiment like that, if you take different types of models of integrated fire type, they know how to select the correct one, which give you the correct output. So the black one is a signal, the red-blue one is a solution of integrated fire, and so they knew how to, that it's a good model, it's a good representation of the potential. And so they decided that now it's easy to write a system of neurons interacting, just like what Boltzmann did is to write a system of particles colliding and interacting by collision. They decide that you can write a system of neurons interacting by spikes. Each time you have a spike, potential grows a little bit, and you get an equation like that, which is again part of probability. It's what we call a stochastic differential equation. This is a part of probability theory. And we know that there is a tool, which is also coming from physics, which is called mean field theory, midfield limit, that is the number of neurons is very big. Then you go from a description of potential of each neuron to the description of the number of neurons with a potential V. So change formalism, come to PDE, and now describes the density of neurons with certain potential. And these objects are partial differential equations, which we know how to handle very, very well now. They have nonlinearities which are specific, which come from there. Even though this kind of object seems to be like the others, surprisingly, we did not know the structure of them. It's only very recently that we discovered that, in fact, these solutions, the solution of these equations can blow up. So there are no global solutions. It might be that there is a phenomena which is surprising, which is that the density of neurons suddenly becomes singular. This is what you see here, the potential, the number of neurons which have a certain potential becomes very, very singular at this point, and solutions stop existing. Standard solutions stop existing. And so when we observed that with my colleagues, we went to see the physicists who have been writing the model and tell them, but your model doesn't have global solution. What does it mean? Our network, our brain works, so there should be something wrong. And so what I said, yes, you are correct. This model is simply what you call a blow-up. When you have all these neurons which spike together, is simply what in the language of physics we would call synchronization. It's a symptom simply that these networks begin to synchronize. And all this spike, all this number of neurons which arrive at the same potential together is simply that they are synchronized, and this is something which we can find in different types of models, which have been used by several people. And so if you now go further and include, for example, refractory states or randomness in the potential, what you find out is that the solution, of course, is very peaky, is very stiff, because we thought it was blowing up. But after regularization, it simply becomes periodic. And so such a network, coming from simple modenic aspects of neurons, has this capacity to create the activity that you record in the brain. Of course, this is a success of this kind of models, that your brain, when you are at rest, has this periodic activity with names that you have heard about them, alpha, beta, delta, which are different frequencies which you record when you are at rest. And this periodic solution, this synchronization, you can observe that, of course, in experiments with the correct shape. Now let's go to another field of biology, nothing to do with ecology, nothing to do with epidemics, nothing to do with neuroscience. Let's go to colonies of bacteria. Bacterial colonies are very interesting because bacteria are very simple living objects that do very few things. They are not very well organized. The cell of a bacterial cell is not very sophisticated. And if you culture them and let the colony grow, so it means bacteria will divide because nutrient is full, then you will see that they will not divide and move like a spherical wave, like something which would be completely random, which would go like a spherical wave. What you observe is that they colonize the Petri dish of this experiment in such a way that they can go very far without being too numerous. Of course, if you had with the same number a spherical wave, it would be much smaller. So these bacteria have invented by evolution ways to invade in an economical way, to find new food in an economical way, the Petri dish. And this is the collective behavior of the cells. So each one is behaving very simple way. The global picture is this. So we know mathematical models which can do that. So this is now a simulation of equations, of partial differential equations which do that. And you see that you create this kind of patterns by a process which is called chemotaxis and using the nutrients. There is another observation related to bacteria, is that now if you put them in a tube, you go to one dimension rather than two dimensions, you see something else which is very surprising for bacteria which are supposed to move randomly, is that they also synchronize in the tube and they move collectively. Here is the recording at time t equals 0. This is the recording at time 2000 seconds. And you see they have been moving collectively with constant speed. So again, something very surprising. How is it possible that cells, bacteria, which just move by swimming randomly, can have a collective motion, which is to stay together and move with constant speed? And this is, again, something you can explain with hands, but you might be completely wrong. If you write equations, you can prove that this is what happens. And if you want to do that, you have to go back to the way cells are moving, and on this bacteria, the way they move is by motion which is called run and tumble, which is very close to what Boltzmann invented for molecules. So these cells, what they do is they just make jumps, and suddenly they change direction. Just small molecules which move with a straight line and then collide, and so they change direction. So cells do that. I mean, this bacteria, this is Escherichia coli, do that modulated by the chemoattractant, which is a molecule outside. So the environment is just a molecule. They react to the molecule by changing direction. According to this molecule, to this chemoattractant. And what you observe, finally, if you observe these cells, are this kind of motion. So each cell moves randomly with this random motion of run and tumble, and if you look at the movie carefully, what you see is that there is a band which is bright. What you see here are a few cells which have been engineered to be very visible. Behind, there are many spots. These are all the other cells which are not engineered, and they make the background. So if you look at the movie, what you see is at the beginning, the bright band is on the middle, and slowly it moves to the right. So the effect of all this random motion together is simply that the bright band, the collective motion, is from left to right. So each one makes a very erratic motion. The output is simply this collective motion that we have seen at the beginning, that the colony altogether finally will move with a constant speed. That's what we want to explain. If we want to explain that, we have to go back to Boltzmann formalism. These are the equations you will find if you write this process. Then you can make simulations, and what you can compare is the solution of these equations that you find at the end with the profile of the density of cells. And you can fit completely the data with the experiments. So this is an example where we know enough the parameters of the motion. We know exactly the time, physicists and biologists know exactly the time of run, the condition of tumbling, so that you will know exactly the parameters. You are very close to physics. You know exactly the parameters, and you can fit completely the mathematical model with the dynamics of the colony. This is again an example where you have different scales. You have a population scale, which is the global motion of the population. You have the individual scale, where each bacteria is moving by run and tumble. And nowadays, the biologists tell us what is the molecular content of these cells. So we have even more complicated models, which are now at the molecular scale, which explain the kinetic equation of Boltzmann, which explains the macroscopic behavior of the cell. To conclude, I would like to give now an example which is going to medical science. One of the places where science is extremely active now, physicists are working on that, in mechanics, people are working on that, in mathematicians want to compute these solutions, it is the growth of tissues. How you can describe in mathematical terms what are the laws of the dynamics of living tissues. And in particular, the question behind is the growth of cancer, of tumors. How do you describe that? And the fact is that a living tissue is quite complicated. It is a mixture where you have cells, of course. You have what is called an extracellular matrix, which are all these fibers which create a tissue, the collagen in the skin, and many other biopolymers which maintain the tissue. You have water, interstitial water, extracellular water. You have molecules in there, different types of cells, of course. So this is a complicated media which is close to what you will find if you want to modelize Earth. Earth is also a very complicated media. If you want to grow plants, where you have water, you have different cells, you have mechanics behind. So the idea is to explain in terms of mechanics how a tissue can grow, how cancer can grow. And these are examples here of observations of experiment of tumors, of cuts of tumors, and these are examples of numerical simulations. And again, physicists and in mechanics, they came to the idea that you should describe that as a fluid which means that you go exactly in the same direction as we said to now. You have, to describe the population density, you have a velocity, you have a pressure now. We are in the field of fluid mechanics. And you have to say how the change in number of cells is connected to their motion, V, and their division rate and their death rate, because now we are in a living domain. We have not only physics, we have also these biological aspects. And then you build up models, and here I used the simplest one, which is to say that you have a Darcy law, because cells are moving in this extracellular matrix like in a porous medium, and you get equations which describe the living tissue. So this is a very simplified model. Of course, there are much more complicated equations behind. One of the main new idea, which I think was was described first by Bernd Drasdo, but the group in Curie Institute and here, by the way, now also was very active on this field. They explain why pressure exists in a tissue. It's not a notion. The notion of pressure in fluid mechanics is well known. The notion of a pressure in a tissue is much more complicated to understand. So they gave an explanation of that, but also they explained that in the Lotka-Volterra equations, the control of the death and the division of the cells is controlled by the pressure, because of the way the cells can sense their neighbors, and they have adhesion proteins which can trigger cell division or stop cell division. Maybe I will not go further than that. But simply what I want to say that these models, again, you can make numerical simulation. You can compare what you get out of them to experiments. So this is an experiment. This is in a cell culture. What you see in this mathematical model is that what you expect is that there will be the cells will come together. It's not obvious in this model that you recover really a tumor as you think of it as a compact object. This is what you see with a free boundary, and that's what you observe in the experiments up to statistical errors to the experiment. You have a free boundary which is moving like that. Of course, when you really want to describe a living tissue, you have to be more complete in the modeling. So here I give an example, which is again a very simple example, where now you say that you have cells, you have nutrients, and you have a pressure. And the nutrients, you can think of them released by the blood vessels, so they are diffused. So you can think of that as the oxygen, which is diffused from the blood vessels to the tissue, and you can create new models which are more realistic, which are closer to what you observe as a tissue. So in this case, what you see is really that you have a proliferating rim plus a necrotic core, which is what you see in real tissues. And you see that also the boundary can be irregular, which is also something you can observe in some tumors. Nowadays, this kind of models, of course, you have to make them more realistic, to include many biology in the models. You have to say that cells can move actively and they are not just pushed by the pressure, as we said before. You have to say that the vasculature of a tumor can be remodeled. You have to say that you have different types of cells. If they don't have enough oxygen, they become quiescent. They can die in what we call necrosis. That you have also healthy cells, of course. If you do that, and if you put all these ingredients and much more of them in a model, what you can do, or what some teams in the world do, is they can take images from patients in hospital, and they can predict the growth of the tumor. So this is these two examples, these two images are computations based on real images of real patients, where they run the mathematical model and check the dynamics of the tumor. And the team which built that is working with an hospital to help medical doctors to take decisions on at what time you have to give the therapy. I will stop here. So what I wanted to show is that now modeling in biology is a domain which has become serious, just like in physics. It's very old, from the 18th century. We know that the phenomena of physics can be described by equations. It turns out that now many examples in biology can be described by equations. Of course, mathematicians are not only interested in seeing equations. They are interested in seeing why the solution exists, why do they exhibit synchronization or blow up, or do they have this pattern formation ability that we have seen? And this relies always on small parameters, going from one scale to another, from molecule to the cell to the organ. So the question, of course, now from a biological point of view, is this useful? So what I try to convince you that now you can really explain the phenomena in mathematics, but you can also do that in a quantitative way. In several examples, not only you describe qualitatively how a neuron is spiking, but you can also fit precisely with experiments, you can fit new experiments with the numerical solution of PDEs. And so this can give explanation. And also I wanted to especially use this case of cancer to show that there are concrete applications which can save life. It's not easy. I don't say that the status now of equations is the same as the question in biology as in physics. It's not true. It's much more complicated. Especially one of the reasons why it's much more complicated is that parameters are not fixed like in physics. Mostly in biology and more specifically in medicine, you don't have access to the parameters. They are not fixed. And the typical example is the speed of light. That everybody knows what is the speed of light. If you learn it. If you want to speak of the speed of division of your cells, you would have difficulties to quantify it. Nobody knows what is the speed of division of its cell. And the reason is that there is no answer. It depends on which cell. So, for example, the skin will renew with a very different time scale than your neurons or your heart or your liver. So all these scales are very different. And this speed of division of your cells will depend if it's during the night, during the day, if you are in good health, if you are tired, and so on. So there is no answer to this question. So of course it's complicated to put that in a model in mathematics. Because it is timely today to speak of innovation process, we heard today that the new case of this virus from Wuhan is coming in Singapore. So I would like to conclude with another equation which describes the spread of epidemics. And this is not new. This is the spread of plague in the Middle Ages in Europe, which was recorded. This is the spread of muskrats in Europe also, which was recorded. We know perfectly, if you can reassure you, we know perfectly the equation which describes that. It has been fit to experiments, so you are safe. So these are my collaborators with whom we have been working that. Thank you very much for your attention.
Well, thank you, Professor Perthame, for a very interesting and I would say enlightening lecture. It gives us, well, certainly to me, an introduction into the role that mathematical equations play in biology. We would now invite the audience to ask questions, if any. Please just proceed with your questions.
Yes.
Hello. The parameter you talk about, that's unknown, right? Can we design this experiment to collect data to learn those parameters?
Okay, so this question is extremely complex. In the case of bacteria, they know exactly the tumbling rate. So they can measure for individual cells the tumbling rate. So in that case, you can measure the parameters. You don't need to access to them indirectly. So this is a particular case. And not only that, but you can also engineer the cell. You can change their genes now in order to change these parameters. So you can do more or less like in physics and do that. In the case of cancer, for example, you cannot change the DNA of your patient. So the only way you can do is what you say. So now you consider that your parameters are distributed, and then you have to reverse, to make an inverse problem to try to find the parameters. So that's much more complicated, and this is one of the big questions in mathematics. How will you be able to run these models when you want to test a large family of parameters? Each of them, in the case of cancer, these are 3D computations. So three-dimensional, tumors are three-dimensional. These pictures are 2D, but beyond that, there is a 3D picture. And that's computationally expensive.
Okay, thank you.
Hello. Yeah, I have a question about you have mainly focused on partial differential equations.
Yes.
What about other things like algorithmically generated fractal-like structures? Do you consider those kinds of things part of?
Yes. That's
What's your opinion on them?
You're absolutely correct. What I did is take the point of view of partial differential equation of modeling by this tool. But again, yes, of course, there are many other tools, especially I mentioned several times probability and stochastic calculus, but also many aspects of informatics are behind this. Of course, yes. All this discrete. Also, one of the bias is always to go to the continuous description, because you have many cells. I spoke of billions of cells for a small number of neurons. So I also take the bias to go to continuous modeling. But all the discrete world is extremely important.
Yeah, I have two questions. First one, it seems from those equations from physics, many equations are linear equations, like a Laplace equation, Schrödinger equation, Fisher equation, they're all linear. There's a non-linear part. But from your equation from biology, seems all of them are non-linear. What's the reason behind?
Yes, you're correct. But let me recall you the slide. Let's go back to this slide. This one is non-linear. The serious equations are non-linear.
Many they are linear, right? It seems
the first one.
From biology, seems no single equation is linear.
No, but I think you have to see that in the history of mathematics. Of course, the first equations which were written in the 19th or the 18th century were linear equations. But from the beginning of the 19th century, you see non-linear equations. Fluid mechanics, Boltzmann, you see non-linear equations. So I think biology comes after with all this background, where you use all this background. People have invented these models of neuroscience because they knew the equations of Maxwell. They knew all this cable propagation of electricity cables by Maxwell. It's not by chance that Hodgkin and Huxley were in Cambridge. Cambridge was the place where Maxwell was. So they had a huge knowledge, one of the best knowledge of electromagnetism in the world. So so they could use it for biology. So biology is non-linear because it's come in a world where we know how to manipulate that.
Okay, second question is, you can see most equations from physics just named by a single or at the most two authors, some two names like that. Your equation from biology, majority no names, because then does that mean they're not well accepted by the community yet, or
no, I think
no names there.
Lotka-Volterra,
The main equation no names yet.
I think they have names. Lotka-Volterra is a name. Keller-Segel for chemotaxis is a name. Hodgkin-Huxley have been. I mean, I think the history now has accepted their names. And there are other names which are also from enzymatic reactions. They have names. So I think that history has been also working a little bit in this in biology, and that these names are accepted.
Any more questions? I do have a question. So all these equations in biology or fireball or biology that were obtained, they were presumably based on observations and experiments, at least for a start. So what would be the size of the sample size before people would come up and say, oh, this is the nonlinear equation that describes cell reproduction, for example?
The problem is that whenever you observe something, you want to explain it. That's the problem of human beings. They want to explain what they observe. And so in physics, like in biology, when you observe something, you want to understand it from the scale below. So if you observe an organism, you want to understand the cells. If you see the cell, you want to understand the molecules, and so on. So there's always this tendency that you want to go more precisely to the next scale to understand what is going on. And so I think there is no answer to your question. When you have a macroscopic phenomena, you are not satisfied with the macroscopic explanation. You want the microscopic one.
So at what point would they say that, based on the observation I've made, this is the nonlinear equation that describes just the phenomenon? I mean, when will they have the courage to say that?
How is it that Hodgkin-Huxley is considered as the correct model for signal propagation? I think that this is when you really have completely fit the data with the experiment, and where you have seen the effect of parameters. So one of the big success of Fitz-Nagumo is not only to have explained the Hodgkin-Huxley, is to have to explain to have compared the signals. It's also that you can change parameters and classify neurons according to these parameters. And then you arrive to a classification between excitatory and inhibitory neurons based on the parameters. And that was exactly what biologists observed without a notion of a way to classify them. So one of the successes has been to classify, to go further than the experiments and to explain what you observe by this model. So I think that.
Okay, yeah, thank you. So if there are no more questions, I'd like to present to Professor Perthame on behalf.
There's another question out there.
Since this is a public lecture, so maybe everybody want to ask you that what is life? I like very biased your point of view. It's okay, PD from point of view, but implicitly I think your lectures suggest a couple of things for me, at least. But I would like to learn from your lecture something about the question, what is life, from your viewpoint.
Oh, you will not like my answer. My answer is life is chemistry.
But you suggested, for instance, multi-scale and hierarchical structures. It suggests something. The part of the not the right question. It's many answers to these questions. But, Oh, maybe. Okay, thanks.
More questions? So I'd like to present on behalf of the Institute a token of appreciation to Professor Perthame. This is actually a coffee mug, but with it is inscribed this Navier-Stokes equations, which you talked about just now.
Oh, okay. Thank you very much. Okay.
So it is said that for mathematician, drinking coffee, one generates theorems. But in this case, I guess from drinking coffee, Professor Perthame will discover many more new equations in biology. So thank you very much for a wonderful talk. Please join me in thanking Professor Perthame.